welcome to Medical Dialogues. I'm Mr. Zaman and today I'm going to talk about genes that may predict complications from obesity exhibit sexual dimorphism finds study. In a recent study, researchers from the University of Chicago characterized the impact of a genetic variant associated with a higher waist to hip ratio, a measure of abdominal fat that is necessary for the formation of fat cells in humans. This variant is also associated with higher cholesterol and triglyceride blood levels in women, but not men, and it contributes to excess fat accumulation as the result of diet in female mice, but not males, suggesting that similar genes might lead to different patterns of fat distribution and obesity-related diseases risk for women. In the new research, the team conducted several transcriptome-wide association studies, that is TWAS, that compared the genomes of hundreds of individuals to identify genes that are linked with obesity and a higher waist to hip ratio. The ratio is what we call a sexually dimorphic trait, meaning that it appears differently in males and females. The genetic analysis found 91 genes that help regulate fat distribution primarily in women. Most of the genetic variants associated with these genes don't alter sequence of the genes that code for proteins. Instead, they occur in non-coding regions that are believed to control where and how much a gene is expressed. Interestingly, most of these variants are in a class of DNA elements called allorepeats or retrotransposons. These so-called jumping genes may have wired fat distribution patterns in humans with implications for metabolic health in women. The researchers next focused on a gene called SNX10, which was most strongly associated with the waist to hip ratio in women but not in men. They found that SNX10 controls the ability of adipocytes or fat cells to accumulate lipids and fat deposits. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.